about the brain here a little bit, and the exciting part is we get to talk about white matter and gray matter and what that all is, and so I can give you a little bit of a better definition and understanding about what white and gray matter is. Okay, so first of all, when we're talking about the nervous system, we're going to specifically keep this to the central nervous system with the brain and the spinal cord here. Right? That's when we're going to discuss gray and white matter. Now, like I said in lecture, okay, gray or white matter is going to be myelinated tissue. Okay, so pretty much, right, well, when you hear me say white matter, you're going to be thinking of myelinated axons. Okay, so when we're dealing with the central nervous system, okay, myelinated axons are referred to as tracts. Okay, tracts are, like I said before, all right, bundles of axon, well, parallel bundles of axons. Okay, so let's talk about what gray matter is then. Okay, that's where there is no myelination. So that's going to be the cell bodies. All right, the dendrites, obviously unmyelinated axons, but also don't forget there's another type of cell that I haven't mentioned, all right, when we're dealing with nervous tissue, and that's the glial cells. Okay, so gray matter includes the glial cells. So when you hear terms like the cerebral cortex, all right, what we're actually referring to is going to be gray matter. All right, but in this case, it's going to be superficial. All right, it's going to be the superficial portion of the central nervous system. A perfect example. So when we were talking about the cerebrum, when we were looking at these pictures of the cerebrum here, okay, all this that we're seeing here on the outside, that's gray matter. So we refer to that as the cerebral cortex. That's like the shell, like an egg, hard-boiled egg. Okay. That would be like the shell. In this case, it's a gray shell instead of a white shell. Okay? And the yolk and everything underneath will be the white matter. Okay? So first of all, you want to know this term. Cerebral cortex is going to be made up of gray matter. Right? It's a superficial layer, like an eggshell. Right? And then there's this other term called a nucleus. And a nucleus is going to be a cluster of cell bodies in the central nervous system. Right, we call that a ganglion. That's mean. I know. You thought you mastered what a nucleus was, but now we're going to call it something different. Right? But in the peripheral nervous system, we call a cluster of cell bodies a ganglion. Right? So the nucleus is going to be this gray matter that we're going to see, all right, inside, which is going to be in the deeper portion of our brain. So the cortex is going to be the superficial, and then the nucleus will be these clusters of cell bodies, gray matter clusters here, that are going to be underneath that surface or deep inside. So I'll show you a picture. I love pictures. Okay. This picture here, this is way better than my old slide because you could barely read any of this. So I know it doesn't look gray here, but it looks kind of like a tannish color. So that's going to be your cerebral cortex, and that's that superficial area, right? Then deep to that, that's where our white matter is. So white matter is just myelinated axons. That's all it is. Okay, but in the internal portions here, you'll see these pockets of gray matter. Okay, and those pockets of gray matter, these clustering of gray matter, that's the nuclei. Okay, we refer to those as the cerebral nuclei. Okay, so just keep that in mind. The cortex is the outer. What's the Cadbury egg, the chocolatey coating or whatever, like the chocolate coating. All right, and then you guys like Cadbury eggs? I don't know what that liquid is on the inside, but that's just weird to me. You know what I mean? No, I know. It's just like Peeps. I don't like Peeps. Some people love Peeps. Well, and especially like my family, they like to let the Peeps go stale. They say it's good. I'm not a huge fan of marshmallow anyway. Oh, really? Okay. I know. I will not touch Peeps. I'm not a huge marshmallow person. But anyways, so that white matter here is just myelinated axons, and the gray matter is going to be the dendrites, the cell bodies, glial cells, okay, and unmyelinated axons, right? And the cerebral cortex is that superficial layer, and then you've got the nuclei, which are deep clusters of gray matter, okay? You can see here, right, in our cerebellum and parts of the brainstem here, okay, very similar. You've got your cortex on the outside, and then internal of that is going to be the white matter. Okay. All right. Let's do a little. All right. Let's talk about the meninges, people. Okay. Okay. This is the rule of threes again. Okay. Remember how the nervous tissue had 3 layers that surrounded the perineurium, epineurium, endoneurium, and then we were talking about muscle tissue. It had the endomyosin, the perimyosin, okay, and the epimyosin. Well, cranial meninges, there's 3 layers, okay? They're all connective tissue, okay? And so they'll encapsulate the brain, okay? And spinal cord. There's 3 layers to them. So in some of those layers, we'll see blood vessels, right? But most importantly, this is where we're going to see the area that circulates the cerebrospinal fluid. Okay, helps to contain it, keep it from leaking out. Okay, because we'll talk about the role of cerebrospinal fluid here in a bit. There's 3 layers, and we're going to start with the deepest layer and go to the most superficial. Okay, the first one is the pia mater. The pia means soft, and so all these meninges, mater means mother, so this means soft mother. Okay, arachnoid, you know what that is, spidery, so that's going to be spidery mother. Right, and dura means hard. This is my favorite. Dura mater, hard mother. Okay, this is the tough one. Okay, so we're going to talk about each of these individual layers here. Okay, so deep is going to be pia mater, and then the most superficial will be the dura mater. Yeah, this is a misspelling. Yeah. Pardon? It should be M-A-T-E-R, mater. Here's the correct spelling. Not mater, like the what is it, pickup truck? What is it, tow truck in Cars? Do you ever see Cars? Is that the right? That's a Disney movie, Car. Yeah, okay. Mater, like in tow mater. All right. Anyways, so the innermost layer, all right, is the pia mater. So this, when you dissect out the brain and the spinal cord, this is that layer that sticks right onto that nervous tissue. Okay, so it's a very thin layer, right? Much like a visceral layer when we're talking about our serous membranes. Okay, remember, this is what's directly on top of the brain. It's made up of a thin connective tissue, that areolar connective tissue. Okay, the middle layer, the layer that sits on top of the pia mater, okay, that's the arachnoid layer. Now, there's a couple important things that you need to know about this layer. Okay, one is the subarachnoid space. You need to know this. This space is found between the pia mater and the arachnoid mater, and that's where the cerebral spinal fluid is located. So when you have to get a spinal tap done, they have to insert the needle into that space. It's no easy feat, okay? But that's where they're going to get the cerebral spinal fluid, right? But also, you have this structure here called the arachnoid trabeculae, okay? And it's this, there's your arachnoid mater, here's your pia mater, and it's this little scaffolding-like structure that extends, right, from the arachnoid mater, all right, to the pia mater, okay? And this is where your cerebral spinal fluid flows, right, in this space right here. So what I'm drawing is the subarachnoid space. So you have these little small structures, these extensions that go from the arachnoid mater, right, to the pia mater, right? So that's how it got its name, because it looks like a web, right? But also, right, that web is mainly made up of collagen and elastic fibers. One, the collagen, as you know, gives it its structural integrity, and then the elastic fibers, well, that gives it that stretchiness there, so it can compensate. Like when you're going to bend or twist, This is mainly, when I'm saying this, I'm thinking more so in the spinal cord, but brain too, like if you're turning and twisting, things have a tendency to shift around, and this allows it to kind of move around a little bit easier, right? So the arachnoid mater is going to be found just underneath your dura mater. Now here's the thing. You have this potential space. Remember, a potential space is a space that is not in existence in a normal, healthy individual, okay? But if there's damage to a structure, right, like the dura mater, blood inflammation can get trapped into that space. And that space can then be occupied by, God forbid, blood, right? If you've ever heard of a subdural hematoma, right, that's when one of the veins in that close proximity has been damaged and it bleeds into that space in between, that non-existent space or that potential space in between the dura mater and the arachnoid mater, right? But just keep in mind, in my drawing here, I'll put, that's awful. Okay, here's the pia mater, here's the arachnoid mater, and the dura mater would be right on top. Okay, so here's your subarachnoid space here. This is where the CSF is flowing. Okay, there's really no space here, but if a blood vessel gets damaged, it'll bleed into this space. It'll cause all sorts of problems. Okay, get really wicked headaches. Now I'm sounding like I'm from Boston. Wicked. All right, the last layer, the outermost layer is the dura mater. Okay, again, dura means hard. This is the hard mother. Okay, it's made up of a dense irregular connective tissue. Normally, okay, it's 2 layers. In most places, those layers are fused to one another. Okay, now it's going to split. Well, we'll get to that in a second here. Right. So the outermost layer, the layer that actually will connect to the bone, makes sense. We call that the periosteal layer. That's going to be the more superficial one. Then obviously the layer that is going to connect to the arachnoid layer, all right, that's the meningeal layer. That's going to be the deeper one. Right? So the meningeal layer is deep, and the periosteal layer is going to be the superficial layer. Okay, and that's the layer that's going to adhere to the periosteum there. Right? So in most places, these 2 layers are fused together. Okay? But in certain areas, they separate and they create a space. Okay? Just like spaces inside your bones, we call those sinuses. So the spaces inside of the dura, we call those dural venous spaces. Now the reason why we call them venous spaces, because your deoxygenated blood, the venous blood, all right, drains into those spaces from the brain. Okay? I'll show you what we're talking about here. So you'll have these spaces, all right, they're not a blood vessel, okay, they're just a space where the venous blood, okay, the deoxygenated blood from your brain, it'll drain into these spaces. Okay? So keep that in mind. Dural, named after the meningeal layer that separates. Okay, sinuses, because there's a space there. Think of sinuses as spaces. And then venous, we're just telling you what's in that space. Okay? All right, and then the last space that we're going to talk about is the epidural space. Okay? Again, this is another potential space. So, in everybody in this room, right, I'm hoping at least, all right, there's nothing going on there, all right? We should have that potential space, right, should be fine, okay? Which means that the dura mater is adhering directly to the skull bone there, right? But in this space, you need to know this is where we see arteries and veins. So if you damage, someone hits you in the head with a baseball bat, okay, you're in a car accident, your head collides with the windshield or whatnot, okay, you could damage the arteries or veins. Then you have what's called an epidural hematoma. Blood will bleed into that potential space. All right, so this next picture here that I'm going to show you will will kind of give you a little bit of a view of all these different structures and spaces and all that stuff. Okay. So here's our portion of the skull, okay, and the brain and everything that we pulled out of it. So let's start from the inside. You can kind of see what I'm talking about. Okay, so here's your cerebral cortex. What is the cerebral cortex? What is that? We just learned it. Yep, gray matter. Right on, right on. It's the superficial gray matter of the cerebrum. Okay, so just on top of that cerebral cortex, this kind of purplish line there, that's the pia mater. If you look, the pia mater goes into these nooks and crannies. Okay? It follows it all over the place. It directly adheres to the brain. Okay, so the pia mater. Now you see there's a little bit of a space here. Then you got another layer. It's like a purplish layer here. That's the arachnoid mater. You can see the arachnoid mater. You have these collagen and elastic fibers that are extending back and forth between the arachnoid mater and the pia mater. You guys see that? Okay, that's the subarachnoid space. It's not potential. Okay, because cerebrospinal fluid flows in there. Okay, so this is where your cerebrospinal fluid is, and you have blood vessels in here. Okay, so then on the other side, here's the arachnoid mater. Then on the other side, you've got your subdural space right here. Okay, now that is a potential space. Okay, as long as everything's fine, potentially there. You know, it's not in existence unless we damage some blood vessels and blood gets into that area there. Okay, then right on the other side of the arachnoid mater, you got your dura mater. Okay, so this is showing you there's two layers to it. The deep layer, that's the meningeal layer. The more superficial layer, that's the periosteal layer, and that's going to line the bone here, right? The periosteum. You can see where the dura mater, the two layers will split off. See how it splits off? You've got the periosteal layer still adhering to the bone, and then the meningeal layer just takes a detour here. It creates a space. And in this space is where venous blood drains into. Anyways, this space is called a dural venous space. Okay? It's naturally supposed to be there. So going back to the dura mater where it's fused together, you have a space here, you can't see it, right? That's the epidural space. That's another potential space, okay? So if you were to get concussed, or I shouldn't say concussed, but someone were to hit you or you were to fall down and strike your head on the concrete, if you were bleeding here, you could get into that space and cause an epidural hematoma, okay? All right, good so far? Yes, ma'am. No, it really depends on just how much it's a blood supply thing, the extent of the damage and whatnot. But yeah, it doesn't take any longer than any of the other ones. In fact, I would say it takes less time because the amount of blood flow to your head, it's quite a bit. Okay, so in case you're not sure, go ahead and check out this picture here. Okay. And just to review, because it's important that you know those. All right. So we talked about dural venous sinuses. Okay. And the dural venous sinuses are where the meningeal layers separate for the dura mater. We have another structure here called the cranial dural septa. Right. So what that is that's sheets of dura mater that are going to extend down into the cranial cavity in various areas. And we talked about this in lab. Was it Thursday? No, Thursday we talked about it. Remember we were talking about the longitudinal fissure, right? That deep depression between the left and right cerebral hemispheres. I was telling you that there's a sheet of connective tissue that will invaginate or extend down into that area. And that sheet of connective tissue is the cranial dural septa here. Okay, so its main function is to support the brain. So the brain's just not jostling around inside of your skull. It helps to keep it in place and support it. Cerebrospinal fluid also helps to deal with that, okay? So it will create divisions between specific brain areas there. Like the tentorium cerebelli was a divisionary sheet between the cerebrum and the cerebellum. We're going to review all that, okay? So the first dural septa is going to be the falx cerebri. This is the one that we saw that is going to divide your left and right cerebral hemispheres. So it's going to be directly midline. It's going to be found in the longitudinal fissure. Okay? Now this falx cerebri, remember, dura mater has two layers. Okay? And when it divides, when those layers separate, it creates a space. Okay? We call those spaces, all right, dural venous sinuses. So the falx cerebri gives us two dural venous sinuses. One is called the superior sagittal sinus. We're going to learn this in lab today, okay? So when we label. And the other one's called the inferior sagittal sinus. Okay? I'll show you a quick picture of them. Come right back. All right. So this picture here is showing a mid-sagittal view of the skull. Okay? So this right here, that's the dural venous septa. What did I say? Yeah. This is the septa, okay? So up here, where the septa divides, that creates this space, and that's called the superior sagittal sinus. Down here, they separate also, and that gives us what's called the inferior sagittal sinus. Okay? Well, so again, I'll go over this with you when we're doing some labeling here. All right. The other one, which I was telling you about on Thursday, is the tentorium cerebelli. Okay? Think tent that is draped over the cerebellum. And this is going to divide your cerebrum from your cerebellum. Okay? It's found in the transverse sinus. Okay? But it is actually, did I say transverse sinus? Okay. You have to forgive me here. My brain's not functioning all the way. All right? It's found in the fissure there. Okay? But it's going to create two sinuses. The transverse sinus, which is going to be found on the back portion, all right, of the tentorium cerebelli. Okay? And then where the two transverse sinuses come together, that's going to form what we call the straight sinus. Again, I'll show you this when we're diagramming. Okay? Well, that's what this is showing you here. Okay? Here's the transverse sinus coming from one side. Here's the transverse sinus from the other. Okay? And then they're going to drain into the straight sinus. And you'll be able to, you can't see it at this angle here so much, but you can see it here. Here's the straight sinus. It's tough to see. When we do the labeling, you'll see much better. All right. Well, in the cerebrum, we saw the falx cerebri. Well, in the cerebellum, we have the falx cerebelli. And this is going to divide the left and right cerebellar hemispheres. Okay? And it also has its own sinus, the occipital sinus. And finally, one of my favorites, the diaphragma cellae. This is what sits over your pituitary gland. Remember, you had the sella turcica, which was like this saddle-like structure, okay, that sat in the sphenoid bone, and the pituitary gland sits down here. And you got the infundibulum that comes up. Well, anyways, you have this dura mater that sits right on top. It's got a little hole in it so the infundibulum can leave. Well, that is the diaphragma cellae. That forms the roof, all right, to the sella turcica. Okay? And that little opening there is for the infundibulum. I tried really hard not to call it the pituitary stalk. That's another name for it, but you guys can't say that. Right? It's the infundibulum. All right. let's talk about the ventricles of the brain real quick. Okay. Remember I was telling you on Thursday that you have spaces inside your brain? Okay. And these spaces, right, is where these oh my gosh, the CSF flows. All right, the cerebrospinal fluid. Okay. And these spaces, these ventricles, are lined with ependymal cells, and it's those ependymal cells that make that cerebrospinal fluid. Okay. So the ependymal cells basically are a filtering system because cerebrospinal fluid is very similar to your blood plasma. And it actually filters out your blood, right? Pretty much keeps the red blood cells out of the cerebrospinal fluid, but takes most of the fluid components from your blood, right? And it helps to make the cerebrospinal fluid, right? Well, it fills these ventricles, these cavities in the brain. And these cavities are all interconnected with one. Okay, so if you look at this picture here, okay, you've got your two lateral cavities. These are these big, long structures here. All right, one is on each lobe, or not, excuse me, each hemisphere. Okay, and then your lateral ventricles will drain down into this little chamber or cavity. That's called the third ventricle. And the third ventricle drains down into the fourth ventricle. That's in your brainstem. And then the cerebrospinal fluid has a couple options here. It can drain down into your spinal cord in the central canal of the spinal cord, or it can flow out, all right, on either side here, and then it will circulate around the brain on the outside, not on the complete outside, but in the subarachnoid space, all right? So I'll explain all that, trust me, okay? So if we're looking at the brain from the front, right, once the cerebrospinal fluid gets to the fourth ventricle, it can continue to flow down here, or it can flow out to the sides here. All right, and I'll talk more about that. All right, so those four ventricles, we have two lateral ventricles, and those are found in the cerebrum. Okay, and we talked about this on Thursday. It divides those ventricles from one another. That's the septum pellucidum. Okay, so the lateral ventricles then drain into the third ventricle, right? And the third ventricle is found deep within the diencephalon. You could basically say that the hypothalamus kind of surrounds it, okay? So this third ventricle sits in the diencephalon, right? And the lateral ventricles drain into it, all right, and the structure that drains the lateral ventricles into the third ventricle is known as the intraventricular foramen. Okay, this little guy right there. Okay, so the lateral ventricles drain down here into the third ventricle. And then the third ventricle is going to drain into the fourth ventricle. The fourth ventricle basically sits, all right behind the cerebellum. Okay, and in front of the pons, right? And it's connected by the third ventricle. The structure that connects the third to the fourth is called the cerebral aqueduct. That's a big, long tube-like structure here. There. Okay, you can see it here better here. Here's the third ventricle right here, and then the cerebral aqueduct drains the third down here into the fourth. Okay. Like I said, that the cerebrospinal fluid has a couple different options. It can continue to drain down into the central canal of the spinal cord, or it can drain out into the subarachnoid space on either side, all right, to bathe the rest of the brain. We'll talk about that. But first, you're so funny. Yeah, yes, we will. No, we will. I just want to knock this one part out, and then we'll get into it. You're so funny, girl. All right. Where am I? Cerebrospinal fluid. Okay. This is a little bit more fun. Okay. Cerebrospinal fluid. All right. A couple important things that you want to know about it. One is that it's clear and colorless, and that's important. Because if they were to do a spinal tap on you and draw the cerebrospinal fluid off, and it's cloudy or turbid, all right, there's a good chance that you have an infection. Okay, so it should definitely be clear and colorless, all right, when it's drawn off naturally. Okay, like I said, the cerebrospinal fluid is made in the ventricles, so it circulates throughout the ventricles and also in the subarachnoid space. You want to know those two areas. More importantly, you want to know its functions. I love this stat. This is one of my favorite stats. Right? It reduces the brain's buoyancy by 90. Not reduces it. It actually makes it more buoyant. But it reduces its weight by 95%. That's significant. If it didn't do that, your brain would pour out of your foramen magnum. I know I've said that to you guys before. Okay? The big function of it is buoyancy. Right? Keeps your brain in your skull essentially. Right? It also acts as a cushion, right, for protection. So it sloshes around, and they're like much like you've seen. I don't know if you've ever read the Cat in the Hat book when the fish is being moved around in the fishbowl and everything. It's sloshing around in there, and that water. Is it high in salt content? Well, I mean, a lot of it it's going to be very similar to your blood plasma. You have a significant amount of sodium in your blood plasma, right? You have a decent amount. I wouldn't say like if you were to taste it, if it would taste salty. You know, I can't tell you that. It's probably going to taste funky. But it's hard for me to say that it's going to have a high salt content. And finally, right, we talk about the environmental stability of the cerebrospinal fluid, right? Again, this is where you're going to think about it. It's literally bathing your brain, okay? So it's going to remove waste material, all right, from the brain, and it's going to sustain it or supply nutrients, all right, for any type of metabolic function, okay? But it helps to keep the brain environment very constant and consistent. That's why. Has anyone ever had a spinal tap before? Okay. So Casey, did you have any headaches after they took the cerebral spinal fluid out? It's not uncommon. And the reason what? Yeah. Okay. So the slightest amount of cerebral spinal fluid, if it leaks out of there, can cause really bad headaches. I don't know how bad basically your migraines were, but I've had people. Oh, okay. So you might have had it. Did you have too much cerebral spinal fluid? Hydrocephaly? Is that why they had to take some out? Sometimes if you have too much, that can cause it too, because there's pressure on the brain. And we'll talk about that. All right, not today probably. Okay, but I know a friend of mine. They did a spinal tap on her. Oh, okay. Well, they accidentally punctured, and she was leaking cerebral spinal fluid, and she said it was some of the worst back pain, headache pain, because it has to be constant. That the pressure in there has to be constant, and even the slightest decrease in cerebral spinal fluid causes really bad headache pain. I really so much want to talk about this, but I won't. We'll wait for next time. All right, let's take a break off of this and then do some labeling. I really do enjoy talking about. Yeah, well, meningitis, and that's the thing, and that's how they can confirm bacterial and viral meningitis is by taking a sample of the cerebral spinal fluid, just to make sure. But to go back to, I don't know who asked me, it wasn't you, Casey, someone asked me about the saltiness of cerebral spinal fluid. There is a higher amount of sodium and chloride in the cerebral spinal fluid compared to blood, but a part of that is because we're kind of concentrating the amount of those electrolytes compared to your blood plasma. All right, so again, not a lot of structures to know for this next part, okay, for the dural sinus and the ventricles. All right, so we're just going to kind of go over that real fast here. Does anyone have a page number they want to kind of call out to me? 50. 80. All right, so in this model here, Right, the falx cerebri, this is, right, a dural septa, okay? So you can see it's this sheet here, right, of the dura mater, okay? It's going to divide your left and right cerebral hemisphere, okay? So keep in mind, right, I'll show you on this other model here. That's the falx cerebri. It's found in the longitudinal fissure. It divides the left and the right cerebral hemispheres. All right. Then we have the falx cerebelli. Now your cerebellum, okay, the two hemispheres will sit down here. One over here, one over here. All right, the falx cerebri separates, all right, the hemispheres of your cerebellum, the left and right hemispheres from one another. Okay. And then this structure here, tentorium cerebelli. Tentorium cerebelli is that dural septa, okay, that is going to lay on top of the cerebellum, okay, and it's going to be found underneath the cerebrum, specifically the occipital lobes of the cerebrum, okay? This divides the cerebrum from the cerebellum. You can see a little bit better, okay, in this picture here, okay? So down underneath the tentorium cerebelli, excuse me, this is where you'd find cerebellum underneath. Okay. So going back to the falx cerebri, where the two layers of the dura mater separate one another, you see this kind of blue here? That's representing venous blood, okay? It's not a blood vessel. That's representing venous blood. It's a space, okay? And in that space is deoxygenated blood, right? So your brain's deoxygenated or venous blood will drain into this space here, and that's called the superior sagittal sinus. And it's just underneath the cranial bone there, okay? So that's on the top portion, right? Down here on the bottom portion, this blue line right here, you can kind of see it, okay? That's the inferior sagittal sinus. So the inferior sagittal sinus will drain into this structure right here. And that's called the straight sinus. Right back here. All right. So the tentorium cerebelli will actually meet up with, okay, parts of the parietal and occipital bone here, okay? That creates its own space, the transverse sinus. This is not the best representation of it. Just label it. I'll show you a better representation of it on the next slide, okay? This right here along this border or margin of the tentorium cerebelli, that is the transverse sinus. You can see it a little bit better here. All right. That is the transverse sinus here and here. And then you can see here the superior sagittal sinus. So this goes up along the top of the cranium there. It goes all the way to the front. Good so far? Not too bad? Okay. So we've seen this before, okay, this view. Okay? This is the longitudinal fissure here, and that's where the falx cerebri is going to be found. All right. Then you got your lateral fissure right here. That's where the tentorium cerebelli will be found. Okay. Now this next model is going to be a little bit scary. Funky looking thing. It looks something straight out of a science fiction movie. The movie's escaping me. Starship Troopers. Does anyone see the movie Starship Troopers? Paul Verhoeven directed it. Okay. Anyways. All right. In case you know what I'm talking about. Those little bug-like creatures. This reminds me of that. This whole model right here that you're looking at. Okay. Have you guys ever seen? I saw a video on YouTube once where this guy takes, I don't know if it was like, it was, I think, liquid aluminum, and he pours it into an ant nest, and then he lets it dry, and he pulls the thing out and then sprays off all the dirt, and it's some funky looking kind of art thing. Anyways, point being is what happened here is somebody took some sort of liquid, right, and poured it into a brain, okay, to fill all the ventricles. Then they just peeled the brain off. And so this, what you're seeing here, okay, what you're seeing here is the actual space. This is all space inside of your brain, represented by this model here. Okay, so you got your lateral ventricles, your third ventricle, and your fourth ventricle down here. Okay, you have this model in your book, yes? Okay, okay. So understand something, okay? This is the anterior view. We're looking at it from the front. So our right is the left ventricle, okay? This is if you were looking at somebody, okay? This is the anterior view. Keep that in mind. Then this model here on the other side, right, that's the right lateral ventricle. So this whole structure from here all the way here, the same here, all the way, all that, that's the lateral ventricle. It looks like from the side, and I'll show you on another model, it looks like a C, okay? Almost looks like a C. Just keep in mind that this is not anatomically correct. We're looking at this person. Right? The next one here you don't have, right? This is the right lateral ventricle. Now this is anatomically correct, okay? So here's the right lateral ventricle, here's the left lateral ventricle, okay? Left and right. All right, you do have this one. Okay? This is the third ventricle. That's going to be, I know, it looks funky, doesn't it? That's the third ventricle right there. Okay? On either side of the third ventricle, the hypothalamus will be found. The intraventricular foramen, that's this structure here. You don't need to know it for labeling purposes. This right here. Okay? That's the passageway that drains the lateral ventricle, right? That being the left lateral ventricle, into the third ventricle. Okay? And then you don't have this one here, but that's the fourth ventricle. This is the posterior view, okay? The fourth ventricle looks like a diamond almost, okay? But you do, here's the lateral view for you guys. Kind of obstructed, but this is part of the fourth ventricle right here. Okay, so the third ventricle drains down into the fourth ventricle. Here you can see the fourth ventricle from your view. This is the anterior view. Okay, there's your fourth ventricle right there. mind you, okay, cerebrospinal fluid can continue down into the central canal of the spine, spinal cord, or can flow out to the sides here. Okay? And then that goes into the subarachnoid space. All right. So this tube here, this guy right here, that's the cerebral aqueduct. That's that tube that connects the third and fourth ventricle. That runs right through the midbrain there. In fact, I'll show you. Here's the anterior view. Here's your lateral view. All right, so now you can see it here in our brain model. There's the cerebral aqueduct. Okay, right here. Space. And the cerebral aqueduct is found inside the midbrain. The cerebral aqueduct connects the third and fourth ventricle. So now, check it out though. I want you to pay attention. This arrow is pointing to the hypothalamus, right? Doesn't it look like it's pointing to the hypothalamus? Except it's saying this is the area for the third ventricle. So I have to caution everybody, right? Make sure when you're identifying this slide, if it's just pointing, right, to that region, okay, of the brain, if it says name the space or the area, they want third ventricle as the answer. Okay? If there's no writing on that slide, you write hypothalamus down. Okay? If it asks for the space or the area, we want third ventricle. Right? If there's no writing on there, then you answer hypothalamus. Okay? Then here, that's the area for the fourth ventricle. All right, we do have a second here, so I do want to go back to the other slideshow and go over cerebral spinal fluid formation. And then I promise you, I'll let you guys go. Right. So that's it for the brain, okay? For the sinuses and the ventricles. Well, they're and the septas, okay? I know. I just want to wrap this up because that way I don't have to smush everything into the next lab. Because, okay, yeah. I don't want to have your brains explode on me, okay? It's just one part here. I really love doing cerebrospinal fluid formation here. Because the other stuff, yeah. Trust me, all good. All right, real quick here, okay? Cerebrospinal formation. You need to know the structure that makes it. That's the choroid plexus. We labeled that last class on Thursday. Okay, but specifically the cell that makes it is the ependymal cells. They line the choroid plexus. All right, so essentially the ependymal cells are going to filter out the blood in the capillaries there. Okay, and it's going to modify, all right, the content of that filtrate. Right? Very similar to plasma, right? But there's more sodium and chloride, but in this case, less calcium, right? And potassium and even glucose. Now, part of that is because glucose is a pretty big molecule, right? So it can't really get into that area there. Now, obviously, all right, these ions here, sodium, chloride, potassium, and calcium, right? Those are much smaller than glucose. So it's a little bit easier to be filled. But for the most part, all right, we're going to see a larger amount of sodium and chloride compared to potassium, calcium, and glucose in there. All right. Okay. So here's the thing that you have to understand. Cerebral spinal fluid is going to be circulated through all these structures continuously. All right. And so it is constantly being made, okay? If you think about it, if it's being made, I told you its content volume has to be maintained at a specific level. Otherwise, if you get too much, you get terrible headaches. If you have too little, you can also have bad headaches and cause problems, right? So as it's being made, you have to get rid of it somehow, okay? And so the extra CSF that we make is going to head into a structure called the arachnoid villi. And I think of it as a mushroom, okay? It's got like a little mushroom cap on top, okay? And as the cerebrospinal fluid comes up, it'll push on the cap, and the cap will open up, all right? And it lets off that cerebrospinal fluid, and it comes out and it drains into a dural venous sinus, okay? So I'll show you a quick picture here, okay? So that excess cerebrospinal fluid goes into the arachnoid villi, and it'll eventually drain into the dural venous sinuses here, right? Which is, where are we? I'm going the wrong way. This guy right here. These structures here are the arachnoid villi. So that cerebrospinal fluid is flowing here. It goes up into the arachnoid villi, and then it drains out. It's a one-way valve. It can't, nothing can go in. Stuff can only leave. So if the pressure starts to build up, no problem. Ever taken a tea kettle and you're boiling water, you know, and you let off some of the steam, it'll stop making a high-pitched noise for a moment because the pressure, you decrease the pressure a little bit. Same type of thing. As the pressure increases here, it shoots it out of the arachnoid villi there. Real quick right here, this is what a choroid, come on, man. This is what the choroid plexus looks like. All right, these are lateral ventricles. So, all right, it's this grouping of cells. So at the center of it, there's a blood capillary, and then the ependymal cells line that blood capillary, and there's a basement membrane here too. Okay, so these cells will filter out, right, the contents of the blood plasma and drains it into this cavity here. Okay, so you can see here, like I was telling you, the lateral ventricles will start to make, right, and some of the other ventricles will be making the cerebrospinal fluid. It flows down into the third ventricle and down into the fourth, right, and then it can either go down in the central canal or it can flow out to the sides, and that's what this is showing you here. It's flowing out into the subarachnoid space here. And so as it's flowing into the subarachnoid space, as the pressure builds up, it'll push the cerebrospinal fluid out into the dural venous with the deoxygenated blood. No problem, okay? Because once that pressure decreases again, this closes down and it locks that out. And the cerebrospinal fluid just goes into the dural venous blood and it gets circulated through your body, and your organs take care of it. Break it down. What they do with blood plasma. All right, that's it. I just wanted to get that out. I can see the expressions on your face. They're like, ah.